Here's how a spectrophotometer works. A lamp provides the source of light. The beam of light strikes the diffraction grating, which works like a prism and separates the light into its component wavelengths. The grating is rotated so that only a specific wavelength of light reaches the exit slit. Then the light interacts with the sample. From this point, the detector measures the transmittance and absorbance of the sample. Transmittance refers to the amount of light that passes completely through the sample and strikes the detector. Absorbance is a measurement of light that is absorbed by the sample. The detector senses the light being transmitted through the sample and converts this information into a digital display.
The purpose of a tunable monochromator is to convert a broad band spectral input into a narrow band output. That is, many wavelengths enter and only one, or a very few, exit the monochromator. This narrow band output can be used to excite a molecular species, as in fluorescence or phosphorescence spectroscopy, to excite an atomic species, as in atomic emission spectroscopy, or actually any number of generic absorption experiments. In an absorption spectroscopy experiment, light that is absorbed by an analyte contained in the sample cell is used as a means of determining the amount of analyte present, usually via a calibration curve. Again, the amount of light absorbed at a specific wavelength reflects the amount of analyte in a sample during the absorption. Although there are many different versions of monochromators, the flavor that we shall examine here is called a Cherney-Turner design. It uses 2 slits, 2 mirrors, and a grating. The light from most lamps contains so many different wavelengths that our eyes interpret this mix as white. That's what white light actually is, a mixture of many different colors or wavelengths. The monochromator takes this white light input and, using a well-machined slit made of 2 parallel pieces of metal spaced very close together, it allows the source beam to fall on the first mirror inside the monochromator cavity. If you could put your eye here at the first mirror, you would see bright white light coming from the source. This mirror's job is to reflect the incoming light, now shaped like the hole from the entrance slit, and collimate that light and allow it to fall on the grating. In older instruments, a prism would be used here instead of a grating. The collimation process makes all of the light rays reflected onto the grating parallel to each other. That is, they do not diverge as they travel from the first mirror towards the grating. If you could put your eye here at the grating, you would again see bright white light coming from the collimating mirror. The surface of the grating has a series of very finely etched ridges or grooves with very specific spacing. This grooved surface is also coated with a highly reflective material such as gold or aluminum, which reflects the light striking it with high efficiency. Collimated light reflected from the first mirror and, remember, shaped in an image of the entrance slit, reflects off of the surface of this grating. Now, an extremely important thing happens. This grating reflection process involves combinations of constructive and destructive interferences of the incoming light rays as they bounce off of the reflective surface. This process separates the many individual wavelengths of light from the white light source into rays of light no longer traveling parallel with each other. Instead, each wavelength is, in effect, reflected at a slightly different angle off of the grating surface and is aimed toward the second mirror. This light separation process is called dispersion and is the heart of what is occurring in this monochromator. Individual wavelengths are separated in space. If you could put your eye at the second mirror looking back at the grating, you would no longer see white light. Instead, you would see a series of closely spaced, colored light bands. The second mirror in this instrument reflects the light from the grating and focuses it at the plane of the exit slit. But since the light rays arriving from the grating are dispersed in space, when they reflect off the focusing mirror, they are again passed on at slightly different angles toward the exit slit. This mirror is designed to collect and focus each wavelength in each band onto a different spot on the exit slit plane. Ergo, when these light bands arrive at the exit slit, they form a series of closely spaced, individual colored bands grouped by wavelength. If you could look at the back of the exit slit, you would see a rainbow of light shaped like the entrance slit. As our movie shows, all red light rays converge on one spot near the exit slit, all greens at another, all blues at a third, and so on. Therefore, when the monochromator is tuned to the blue wavelength, say 425 nanometers, the blue light rays are focused onto the slit itself, and all other wavelengths strike the inside wall of the monochromator around the exit slit and are absorbed. This is why monochromators are internally painted black, to absorb this and any scattered light flying around inside the monochromator cavity. Finally, notice that the photomultiplier tube, or PMT, sees only blue light that is passed through the sample cell from the monochromator. The second tuning step in our movie shows a very slight adjustment of the position of the grating, in modern instruments controlled by a computer, and therefore a slightly different reflection angle of the light rays striking the grating. This translates into a slightly different position of the wavelength bands at the plane of the exit slit. In this case, the monochromator has been tuned to allow 500 nanometer, that is green light, to pass through the exit slit, and all other wavelengths, including the blue 425 nanometer light from before, to be blocked. As can be seen, the PMT now detects the 500 nanometer light's intensity. And again, analyte content will be determined by a calibration curve that plotted absorbed light at a specific wavelength versus known analyte concentrations.
Phototubes and photomultipliers. In this video, we'll be discussing two main points. The first main point is, what is a phototube and how is it used? A phototube is a device that controls the flow of electrons by means of fluctuation in different wavelengths of light. It is designed to emit an electric current when exposed to light or any other form of electromagnetic radiation. The phototube has earned the moniker electric eye for its use in a wide range of light sensing applications. These applications include spectrophotometry, blood analysis, and pollution analysis. We'll take a closer look at spectrophotometry and how a phototube is used. This is a diagram that shows a phototube is connected to a primary light source. The light is then emitted to one single wavelength of light through the use of a monochromator, which passes through the sample cuvette and then unabsorbed light then passes through to the detector. In this case, a phototube, which then converts photons of light into electrons, can be read through a data collecting device. The phototube works by the principle of the photoelectric effect. This diagram illustrates the principle of the photoelectric effect. First, photons of light are emitted from a light source at a certain given wavelength. The photon then strikes the photocathode. The cathode is coated with a bialkali coating which ionizes the photon, creating electrons that are then collected at the anode. The anode collects these electrons, which creates an electrical current through the system, and then we are able to use that information to collect data. We will now discuss the different parts of the phototube and their purpose. First, the glass casing allows to house the cathode and the anode, and allows to reflect any ambient light that may be present. The photocathode is the primary site where the photoelectric effect initiates. Again, through the coating of the bialkali substance on the cathode, it converts photons of light into excited electrons. The anode is the site where ionized electrons are collected and then transported to the readout device.
Our second and final point will be to address what sets photomultipliers into its own category and its usefulness in the amplification process of converting photons of light into usable and more functional units of current. A photomultiplier is a modification of a simple phototube. It is a special kind of phototube, being that it is extremely sensitive to light because of its ability to amplify. Photomultipliers are used when light fluxes are extremely low. A high light flux could easily damage a photomultiplier. This is another reason why photomultipliers are able to amplify at such low light intensities. The mechanism of a photomultiplier starts out when a photon is first emitted into the cathode, which is then converted into an electron. The photocathode is coated with a bialkali coating which ionizes the electron and is then passed into a sequence of diodes. Each diode is coated again with the bialkali metal that initiates the photoelectric effect. This process is then able to turn a single electron through a series of interactions with the diodes into a million subsequent electrons. Once electrons have successfully gone through all the diodes, it is then absorbed by the anode and sent to a readout device, creating an electric effect.
In conclusion, phototubes are used to measure the changes and fluctuations in different wavelengths of light. This is done by the photoelectric principle. Through this principle, photons are able to be ionized through the interaction of the bialkali coating on the photocathode. Lastly, photomultiplier tubes are used to amplify wavelengths of unabsorbed light through the same principle, but with the addition of a series of diodes that can amplify even the smallest amount of electron signal.
Hello, I'm Chris Roberts from Park Avenue Gold Exchange, and today we're here in the lab and we're going to be testing gold. I'm going to show you how we test gold versus our competitors. We use a spectrometer, and this device here is an X-ray device that will X-ray each item that we place in there and tell us the complete metallic composition of the item. So I'm going to place a bangle in here now. And within 10 seconds, we're going to get the complete breakdown of this item. So in testing this item, you can see it has just a touch of iron, a little bit of cobalt, 29.9% is copper, 6.29% zinc, silver is just under 5% at 4.67%, and this is testing out at gold, which is AU, at 14 karat.